anyone can ask questions. We can sort of go through all those questions. When those are done, then we'll have everyone log off. And then his committee will be there to grill him and ask him lots of hard questions about his thesis. So that's sort of the goal. All right. So, it's kind of funny, I was thinking about it last night, like what I was going to say about Juan when I got out here. So Juan's been in the lab for a while. I mean, he started in UT Austin. We moved here. We've been here, I guess, about 4 years now. When he started in the lab, I think like a lot of the grad students, I let him search around looking for projects. And so Juan, I think what you'll see today is Juan's had a diverse number of projects that he's actually worked on. And I think a lot of this came about because Juan was kind of figuring out I think a lot of what he was interested in early on, and he participated in a lot of different things. And usually in my lab, I think it's about 3 years before people have enough sort of background and they've developed something I think really useful that they can start applying. And I think what you've seen from Juan is, and I think what you'll see today is he's gotten a lot of data in the last couple of years. But I guess the one thing that I would say, and I think that would impress upon you is, what you're going to see today, is Juan's done some genetics. He's done 2 different animal models. So he's learned to do a fair number of animal experiments. He's actually developed methods to actually image from animals. This is actually really hard. I know you guys are going to see some really pretty images, but I just want you to, when you think about this is really hard work. And Juan will tell you hopefully a little bit about why it's really hard, but this has not been easy projects, just learning how to image tissue from animal models. Juan's also participated in some human studies. And then he's also done probably a lot of what he's done is sort of develop some computational techniques so that he could quantify spatial structure. So he's had a pretty broad, I think, education, and that's what I was hoping for him. He's had a lot of experience. If he actually passes today, which I hope he does, then he actually he was offered a postdoc at Vanderbilt from Eric Scarr and Jim Cassatt in their labs. And this is a pretty competitive postdoc. They only hired one person for this new position. And this new position is you're going to come in and use not just microscopy, but a lot of these sort of techniques to really move in and think about trying to understand infections in a new way, using imaging and mass spec and some of the other things. So looking at kind of chemical gradients and other things in infections. So I think that's about it. And Juan, I'll let you go. And yeah.
Cool. Awesome. Thank you very much, Marvin. Really kind introduction. And thank you everyone for being here today. I'll talk to you about some of the work that I've been doing for the past few years. And the title of the talk is Characterizing the Micron Scale Spatial Structure and Physiology of Bacterial Communities. So it's basically microbial communities, how they arrange in space, and what does that mean for infection. So one of the main thing that really sparked my interest in biology when I joined grad school was microbial communities and how some of their functions influence so much of human life, from delicious cheese and wine that we consume to ultimately infecting these infections that some of us succumb to. And so if you look up the definition of a community, is a group of 2 or more species in a common location. And this location is primarily the environment. And what I've learned is that the environment is a fundamental part of the interactions within a community and ultimately the function of a community. And so here's a picture that I took on a infection of a microbial community. In red is one kind of bacteria, and in green is another kind. In blue is primarily the host cells. And what you can see from this image, we can start to interrogate the infection. And so the first question would be like, well, who is there? And this is fairly easy to do. Now we have a lot of tools to characterize this, to answer this question. We have 16S RNA sequencing. We have FISH, which is a technique to label specific parts of the genome of a bacteria that will identify itself. And experimentally, so in this case, I know which bacteria I put a brick green bacteria that fluoresces in green, one that fluoresces in red, right? So the next question is a little bit trickier, which is what are their functions? So what is this community doing? And in the Wiley lab, we use RNA-seq to try to answer this question. And work here also use mass spectrometry to get into understanding this function question. So what is going on in the community? And so when I joined grad school, we started answering this other question that we don't really know about, which is where are they, particularly within bacteria themselves and at the micron scale. So what is the spatial structure of microbes in an environment of interest? And this is what my thesis has been about. Like, we know who's there, we have a pretty good idea about what are they doing, but we haven't really looked at the spatial structure. And while you can always image these things, what I really want to do is quantify the spatial structure. And so my thesis is quantifying the spatial structure in microbial communities. So this is a lot of what I've been thinking about. So you might ask, well, why we want to know about these things? And is spatial structure important? Well, we actually have some pretty good data that shows that this is important. We know this is the case because we've done some experiments in mice, and we collected some human samples. In the case of the mouse, there was this previous graduate student in the lab who infected bacteria, used bacteria to infect mouse using an abscess, and he knew how these bacteria interacted. And the results of this paper show that the spatial patterning of these two bacteria affect their virulence during infection. And this really set the groundwork for my thesis. And in a more recent study where I was actually involved in, we went into humans and looked into cavities. And so this is an image of plaque in a human tooth. Each microbe is a different color. And you can clearly see that there's some spatial organization in this community. This is a biofilm growing on a human tooth, and you're looking down into it. And it colocalizes with caries. And what we learned, we were able to show that this spatial structure is critical to cause caries. So at least here are these two instances where spatial structure at the micron scale is important for infection. And so now we know that spatial structure is important, but how do we find mechanisms that control this thing? Well, to tackle this question, I've used several kind of models several different models to answer this. But ultimately, what I want to remind you is that we want to understand human disease. However, human samples are quite hard to obtain. So we use different models. First, I'll talk about in vitro models. These are models that you use in the lab, and typically involves solutions that you can get a hold of during the lab. And in the Wiley lab, it was developed the cystic fibrosis sputum model. And so this will incorporate part one of my talk. And then the second kind of model, the second class of models is animal infection models. And the goal of these models is really to start understanding the interaction between the host and the microbes there. And during the talk, I'll talk about a surgical chronic wound model. And so the reason why we're assessing the spatial structure in a lot of different environments is to see if it's consistent or not, and try to understand the impact of the environment on these interactions. And so now I'll start with part one, which involves two microbes that I'll talk about, Pseudomonas aeruginosa and Staph aureus, and its spatial structure in vitro. And the rationale is that we wanted to develop a system first to study the spatial structure of microbial communities. So I'll introduce what cystic fibrosis and this model is. So cystic fibrosis is a genetic disease. It affects around 30,000 people in the US. It results in reduced mucus clearance, and it ultimately promotes the chronic infections at the lung. And so the way that based on that, it was developed the synthetic cystic fibrosis sputum, which I'll refer to as SCFM2. And this was developed under this really simple idea. You go out and take an environment, in the case the cystic fibrosis sputum, and then we ask, well, what's in it? So we did that and figured out what's in it, and then we basically made it up in the lab. And it contains organics, salts, lactic acid, DNA, lipids, and mucin. And so we have this environment. Then who are the members of the community? Well, the model community is based on Staph aureus. This is a gram-positive, non-motile facultative anaerobe. It is the most common pathogen in cystic fibrosis, and we know that there's a high abundance of antibiotic strains. This is Pseudomonas aeruginosa too, and it's a gram-negative motile facultative anaerobe. It's associated with decline in lung functions, and it forms aggregates in mucus. So why are we doing these organisms? Well, we found both of these together in different infections, including the CF lung and the chronic wounds. Particularly at the clinic in Emory, you find that about 50% of the patients have both Pseudomonas and Staph simultaneously. And the other reason is this is a good place to start because we know a lot of the ways that these microbes interact. And so how do we know this is a good model? Well, we know that that SCFM2 has similar physical properties to human sputum. We also know that supports aggregate formation of these major pathogens. And the picture in here shows it's a sliced micrograph of human sputum, and in red you find bacteria, and in blue it's just generic DNA, which is associated with hosts. And from previous papers in the lab, we know that the gene expression of these major pathogens, primarily Pseudomonas, mimics that of the expression during human CF infection. And so I told you that now we're going to dig deeper into how do we know these two are interacting. I like to mention that a lot of this, at least most of these interactions, have been done using in vitro systems. So one of the first one is that Pseudomonas influences the persistence and virulence of Pseudomonas aeruginosa. In turn, Pseudomonas promotes toxin production in staph. Additionally, we know that Pseudomonas inhibits staph by lysing it and down-regulating respiration. And what I'm coming to talk here is like, well, what is the impact of spatial structure among this interaction? How is it modulated? And so one of the main things that we know from these two microbes is that they don't really like coexisting in vitro, but we still find them coexisting in vivo in a lot of systems. So these are the main questions that we're going to ask. Well, do Staph aureus and Pseudomonas coexist in SCFM2? As I mentioned before, these two microbes don't really like each other. Pseudomonas kills staph under most laboratory conditions. The next one is, what are the mechanisms driving the spatial structure of this community? And so, well, how am I going to answer these questions? Well, primarily, I'm going to use a confocal microscope. It's an LSM 880 Iriscan. And a confocal microscope, the cool thing about it is that there's a way to generate images in 3D by scanning, by taking slices of an image, and ultimately you build the 3D model of it. And then I built a framework for quantifying the spatial structure using these three-dimensional images. So I'm going to walk you now through the experimental setup of this in vitro experiments. So first I have Pseudomonas and staph. They're fluorescently labeled. One is green, one is red. And then I put them in SCFM2 at 37 in these little cool little chambers. The cool thing about them is that you can image through the bottom of them. And so now we have them under static conditions, and then we grow them, and at the end I collect the CFUs. The static condition is one that has been used in the lab before to characterize their physiology. But I also wanted to know the impact of the spatial structure. So what I did is I added a little stir bar, and then I mixed them and see what was the impact of it. And what I found is that in this graph, so at the bottom, the x-axis is time in hours. So I go up to 16 hours. And in the y-axis is the CFUs, which is colony forming units, which is a proxy for growth, bacterial numbers. And you can see that over time, the dashed line is staph and the solid line is Pseudomonas. And you see that they coexist consistently up until the 16 hours, which is at about the time that they stop growing. The red line represents growth individually and not in co-culture. However, when you mix them, you find that while Pseudomonas does really well, staph crashes and it gets killed by Pseudomonas under mixed conditions. And we were wondering, like, what might be the mechanism for this? And so what we did is, in this case, I grew Pseudomonas by itself statically, and then I used the supernatant, the soup that comes out of it, and then I spotted it in a plate that had a lawn of staph. And what I found that the zone of inhibition was pretty small. And when I grew Pseudomonas mixed by stirring it, I found that the zone of inhibition was pretty large. So there's something about stirring Pseudomonas that makes it more deadly. And we know that some of those killing activities are modulated by oxygen. But mostly what I found is that now we have this model, and this model did just as well in mono and co-culture. So now we can quantify the spatial structure of this community. And so I'll briefly describe what is the framework for this. So in this framework, what I want to be able to do, what I wanted to be able to do is first to identify each bacterium, which one is which. That's really easy. I labeled them red and green. And later on in humans, it's not that easy, but I also want to be able to quantify their biomass, like how much of them they're each. And then the next question is, we'll classify their biomass as planktonic, also known as free swimming bacteria, and or as aggregates. And these two modes of growth or life history are fundamental in their physiology. It's been associated with motility, antibiotic susceptibility, amongst other things. And last thing, I wanted to get some quantitative spatial relation data. And so in order to do this, I developed this segmentation pipeline in MATLAB, which is to identify each bacteria. Then I use some statistical analysis tools and ultimately build a package in the software R that characterizes the spatial structure. Now I'll walk you through the quantifying the spatial structure. And these are some of the images that I took in vitro. At the top in AB, you can see staph by itself. Then in the green is Pseudomonas by itself. And at the bottom, it's both staph and Pseudomonas. And when I imaged these communities, I ended up imaging about 10% of the whole volume of the well. So just to capture as much as I could possibly do. And now I'll show you some of the results in regards to planktonic versus aggregates. And so I found first that in regards to Pseudomona, most of the biomass in both mono and co-culture, in here, monoculture, which is Pseudomonas by itself, is in black, and then in co-culture with staph is in the gray. And so you see that they're fairly equal. I didn't find any significant difference. The other thing that you can see is that the variance is pretty high. This is the standard error of the mean. And this is something that you could expect, since this spatial structure is something that is an emerging property. So these bacteria aren't synchronized or anything. And then we have staph aureus. And in this case, we have that the monoculture had a substantial amount of its biomass as an aggregate. But when you go to the co-culture, it's not as much. So in here, you can see that Pseudomonas is influencing the aggregation of staph, and it's driving it into planktonic growth. Now, I'll walk you through the quantification of the micron scale spatial organization using proportional occupancy. And what I do is I basically go around the image, and then I focus on a part of a bacteria. And let's say in this case it's staph. And what I what I want to see is how its environment in relation to other microbes surrounding it. So I start building concentric circles or spheres in the case of 3D. And I ask, well, who's around it? What is the proportion of the volume that is being occupied by certain microbes? And so in this case, you can see that 20% of the volume is occupied by Pseudomonas, 40% is occupied by staph, and then we have 40% that is just empty space. And then I can take another concentric circle at a specific distance, and then we calculate the same, and you can take step size increments and then characterize the whole space around this. And I do this in multiple places around the image so that I can get a sense of this global spatial organization of the community. And now, in order to go a step beyond, what I started calculating is the enrichment distance. And the enrichment distance is the distance from the focal point at which the proportional occupancy is the highest. So in this case, staph is aggregates. So the enrichment distance is one of the closest distance that you have, but then the highest proportional occupancy of Pseudomonas is a little bit farther. So this is how I characterize the spatial organization. And now I'll go over some of the results from our in vitro experiments. And so as if you compare staph to staph, you find that it is very close to each other. This is about as close as the algorithm is able to tell. And this is easy because we know that staph primarily grows as aggregates. And then what we find is that if you compare staph to Pseudomonas around it, we find that they not only are coexisting, but they're actually pretty close. They're within 3 microns of it. So now I developed this framework where they coexist and quantify their fitness and life history, and now this is the spatial relation. And then I wanted to get to the question, well, what is driving this? What is the role of single molecule that we know that they interact plays in this spatial structure? And so in order to understand what is driving the system, well, we know that Pseudomonas releases several factors that inhibit staph aureus. Amongst them is this molecule called 2-heptyl-4-hydroxyquinolone N-oxide, or HQNO, and we know that Pseudomonas uses the PQSL gene to make HQNO. So what I did is, and we know that HQNO blocks the electron transport chain in vitro models and inhibits respiration of Staph aureus. So what I did is I used genetic tools to remove the ability of Pseudomonas to make HQNO by deleting this PQSL gene. And now I'll go over some of the results of using this mutant. And what I found is that when I grew them together in these chambers, they still coexist, very similar to how they do with the wild type in static conditions. And under mixed conditions, I saw that staph is still being killed, but by not by a lot. In the wild type, it was being killed more. And then in regards to the aggregation versus planktonic, we still see that Pseudomonas is driving staph into more planktonic biomass, just like we saw in the wild type. So next, what I wanted to quantify is like, well, can HQNO, the production of HQNO, impact this micron scale spatial organization of the community? And so what I did is I run the same experiment, run the same pipeline, and I found that, again, if you compare staph to staph, they're pretty close to each other. However, when I compared staph to Pseudomonas, this PQSL mutant that cannot make HQNO, I found that they were actually found farther away from staph. Now, this was not expected, and this was pretty surprising because naively, I would have thought that if you remove a molecule that inhibits the growth, then you would be placed closer together. So, well, why is HQNO the mutant farther away from the wild type? And the answer is, well, I don't know, but we have a pretty good idea. We have some guesses. One of them is that they're competing for oxygen. And what this would mean is the way that HQNO inhibits or staph is by downregulating respiration. However, if you remove HQNO, then staph might start respiring, thus competing for oxygen, and so Pseudomonas might choose to move away in order to limit competition. And so this allows us to address these questions and know about the interactions that HQNO seems to be having. And one of the other things that we know that Pseudomonas does to staph mediated by HQNO is this antibiotic susceptibility, which is ultimately clinically very relevant. And what we know is that it's been found that HQNO protects staph from tobramycin, this antibiotic. And I wanted to test this using this system. And what I found was actually pretty interesting. So in the x-axis, we have staph grown in monoculture by itself. Then we have staph grown in co-culture with Pseudomonas aeruginosa, the wild type, and then we have it growing with the PQSL. And in here is how much staph was collected after the experiment, right? And then in the black, we have the control, so no antibiotic treatment, and in the gray, we have the antibiotic treatment, right? And so what we found is that actually staph by itself does better than both Pseudomonas conditions. However, if you remove the ability of Pseudomonas to make HQNO, staph does poorly. So now we found a system where actually they grow up to the same level under normal conditions. However, when you have an antibiotic challenge, you see this difference in fitnesses, which may extend to a different kind of interactions. So to conclude with this part of the talk, we can say that, that these two microbes, Pseudomonas and staph, stably coexist, grown statically in this host-like environment, that microbial communities are spatially structured through an active process, and that the removal of these interactions may lead to this unexpected result. And finally, that this spatial organization may play a role in this physiology, including the antibiotic susceptibility. So now I wanted to test this new environment, which is the chronic wounds, and it's this is part 2 of my talk, which is Pseudomonas aeruginosa spatial structure during infection in an animal model, chronic wounds. And the rationale of this is we primarily wanted to investigate host microbe interactions. And this model is the chronic wound. So this is a model over chronic wound infections. These infections are defined as wounds in humans that last for longer than 6 weeks, and this primarily occur with immunocompromised people or comorbidities, right? Like diabetes and other stuff. And so the model consists of surgical incision. We use a mouse, and then we make a surgical incision, a circle on the back of the skin, and then we cover that with a semi-permeable dressing. And then we infect the microbes under the dressing, but over the wound. And so we layer that basically on top of it. And then the dressing prevents the wound keeps contamination away. And so we collect these experiments after 4 days. And so that's the model. And so particularly, we're going to do basically the same thing that we did. We're going to infect the mice in mono and co-culture with Pseudomonas and staph. So I'm going to use Staph aureus, Pseudomonas wild type, the Pseudomonas aeruginosa mutant, the PQSL, and then I'm going to introduce another mutant, which is the PHC12, which produces phenicins, and this is another weapon that has been characterized in vitro that affects Staph aureus. And then for data collection, I'm going to use STAPI in this case. Now we have host cells, so we can visualize them too and image them immediately. And I'll also collect CFUs, which is collecting the growth of bacteria. And this work was in close collaboration with Carolyn. She was a postdoc in the lab, and now she is a faculty member at Oklahoma. And then whenever I show data that she collected, I'll just show her picture. And this is how the model, the wound model works. And then this is, I'm just going to show you some pictures of how the chronic wound looks like. This is the wound mounted in the microscope. I wanted to take a picture of where the objective was so that I can get a sense of where am I in location to the wound. As Marvin mentioned earlier, this was not easy, and it took a lot of tweaking in regards to what kind of mountain do we use. But the idea was to make sure that we are imaging the wound in minimally disturbed environment. So we literally just took it, put it under a mountain that is supposed to prevent fluorescence decay, and then image them right away. And so this is one of the images that I showed you before, and this is Pseudomonas and Staph. So Pseudomonas is in green, and Staph is in red, and then the blue are host cells. And so, well, what is now we can go through the data, the results, and what is the impact of co-culture on distribution of biomass during infection. And so now I'll show you some bacterial numbers per sample that Carolyn collected. We found that in regards to Pseudomonas, the purple is in monoculture and the pink is in co-culture, and we found that there's a slight decrease in fitness in Pseudomonas when you put it with Staph. In the case of Staph, we didn't really see any significant difference between them. Then I wanted to ask, well, what is the effect on the planktonic biomass? As I mentioned, the free-floating cells. And we found that both Pseudomonas and Staph were primarily existing as aggregates in both mono and co-culture. This is in contrast with the finding in SCFM2 that the presence of Pseudomonas would promote planktonic growth in Staph. And so then we ask, well, that was the data in the wild type. And now I wanted to ask, well, what is the impact of HQNO and phenicins? And what we find is that first I'll go over Pseudomonas. So Pseudomonas, the wild type, the PQSL mutant is in green, and then the phenicin is in purple. And we found no difference in the growth of either of the mutants. And then when we look at the growth of Staph in co-culture with all of these mutants, we also saw no significant difference in their fitness. I'm missing Carolyn's picture, but this is also collected by Carolyn. And then when we look at the planktonic versus aggregate growth, I'm showing that their Pseudomonas, both of the mutants increase a little bit of the planktonic, but this wasn't statistically significant. And in return, Staph didn't really care if HQNO or phenicin was there. And so I want to catch up, take a breath, and then talk to you about what we've found so far. So in this case, we found that both SCFM and chronic wounds support this coexistence of Pseudomonas and Staph. As I mentioned before, this is not the case in most of the laboratory experiments. We find that Staph biomass in co-culture doesn't shift to planktonic in chronic wounds, but it does in SCFM. And we found that HQNO doesn't affect fitness or aggregation. But what about the spatial structure during infection? And this was actually pretty cool. So what I found is that if you look at Staph surrounding Pseudomonas, they're pretty far away. They're 22 microns apart. And so if the analysis in this case goes, I'm characterizing it up to 30 microns. So this really tells you that there's some segregation of Pseudomonas and Staph in the chronic wound of wild-type Pseudomonas. However, whenever you look at the PQSL mutant, it is Staph is much closer to Pseudomonas. In this case, they're around 13 micros. And in the case of the phenicin mutant, I didn't really find any significant difference, but it seems to be also farther away. And so Now we know that HQNO is impacting the spatial structure of the community in SCFM2 and in chronic wounds. And then the last and coolest part of the experiment is how it impacts antibiotic susceptibility, which is ultimately the clinical relevance of our system. And so this is an experiment that, again, Carolyn did. And so right now I'm showing you the bacterial numbers of Staph aureus after going through chronic wound infection. And so this is untreated. So they were just put in saline solution. However, the next condition was treating them with tobramycin and then homogenizing them. So what she did is basically take the chronic wounds, put them in vials with little metal beads, and then just shake them up so that you essentially remove the spatial structure of the environment and then apply the antibiotic. And what you find is they pretty much, Staph doesn't care if it's by itself. It is in co-culture with with Pseudomonas, the wild type, or the mutant. However, if you take the wounds and take them intact and then just do the apply the antibiotic and then collect the growth after that, we find that in the case of the monoculture, it does better. Then Staph does better by itself. Then in co-culture with Pseudomonas doesn't do as well. And in the case of the co-culture with the PQSL mutant of Pseudomonas, it does even worse. So in this case, we again see this interaction of antibiotic susceptibility and spatial structure impacting the community. And so from this, I can draw some conclusions over the overall project. In this case, we found that Pseudomonas aeruginosa and Staph stably coexist in chronic wound infections. We can see that microbial communities are spatially structured through an active process in this case. We saw that HQNO, but not phenicins or piacyanin, they modulate the organization during chronic wound infections. And we can see that antibiotic susceptibility of Staph aureus is associated with both spatial structure and HQNO during these chronic wound infections. And so now I want to sort of compare and contrast SCFM and chronic wounds. These are two different environments. And at the beginning of the talk, I told you one of the interests was characterizing these shifts in the environments for the resulting from the behavior of microbial communities. And so some of the similarities between SCFM2 and chronic wounds is that we found that both of these microbes coexist in this environment. We know that HQNO was able to modulate the spatial organization of the community. And then we also knew that HQNO modulated the antibiotic susceptibility of Staph aureus. This was what was in common of SCFM2 and the chronic wounds. However, some of the differences that we found is that Pseudomonas aeruginosa increases Staph aureus planktonic lifestyle in SCFM2, but not in the chronic wounds. In the chronic wounds, Staph was essentially blind in respect to planktonic growth to Pseudomonas. And we also found that the enrichment distances are different, as we found even if you compare wild type Pseudomonas and Staph, those distances in the chronic wounds, Pseudomonas was way farther away. And primarily, this was this really interesting effect of HQNO, where it has the reverse effect of bringing together these cultures in the chronic wound, but drifting them apart in the SCFM2. And so these are the projects that are essentially done. Now I can talk about, I'll talk about two projects that are particularly not finished. And one of them is investigating the spatial structure in chronic wounds beyond the micron scale. So this is something that we're starting to look at, and we don't really know how it's moving, how it's gonna work. But the idea is that we want to look at this sort of the macro scale, quote unquote, of the microbial communities. And it's basically taking advantage of these really, really large images that I was able to take from the chronic wounds. So this is 600 microns by 600 microns by 200 microns. And one of the interesting characteristics is that chronic wounds, some of the work that has been done in the characterizing the spatial structure of them is in regards to the tissue depth. And so at the bottom, we have the surface, and deeper, as you go to the top, it's deeper into the tissue. And we want to find what are some patterns that may arise from this interaction. Additionally, I primarily talk to you about inhibiting interactions and trying to interrupt these inhibiting interactions between Pseudomonas and staph to see what spatial organization and spatial structure arises from the interruption of these interactions. However, there are other family of interactions that are pretty pretty common, and we've identified also in lab, and this is in regards to cross-feeding. And cross-feeding is whenever a microbe releases some sort of nutrient, and then another microbe in the community consumes it. And so this is in case of L-lactate, and we have some preliminary data that this L-lactate, which is the nutrient, modulates the spatial organization during an abscess infection. So we are doing the same thing, observing microbes during infection, and this is a picture of them. These images are even harder to get because these microbes are oral microbes. This is a different kind of microbe, and they the reporters aren't as strong, and these microbes don't grow as fast or as well as Pseudomonas and staph. So this has been a little difficult. And what we found is that actually we have some preliminary data showing that if you're able to cross-feed, that is to get nutrients from another microbe, you are found closer together than if we interrupt and negate the ability to cross-feed or to get nutrients from another one. So this is sort of like a fundamentally different mechanism of interaction, and yet we're starting to interrogate the community spatial structure to see if we can see them, these interactions happening. And then I'll talk to you about briefly some other projects that I worked on that were pretty cool. So the first one is in cavities, and I alluded to it on one of the first slides in the talk. These was collaborations. I didn't took these pictures. They were kind enough to send them to me, and then I was able to play with them. So the first one resulted in a publication that was published, I think, last year. And then the next one is we were trying to investigate what is the difference between different kinds of lung infections, particularly acute versus chronic infections, and see if we can see this signature of infection. And this paper is being resubmitted. We got some feedback from it. And so with that, I want to leave with some final thoughts. Basically, what factors influence the spatial structure in the microbial community? Well, it turns out that the environment plays a big role in the microbial community structure, but also the specific interactions between microbes, which is what we explicitly try to measure. The other question is, well, how relevant is this? How is this important? Well, the antibiotic susceptibility to me seems to be really important because this can be modulated by spatial structure. So this is what we are looking at right now. And finally, we can start to use spatial structure characterization to identify interactions within microbial communities. So in this case is using these tools that I built to identify potential phenotypes amongst members of a community. And with that, I would really like to thank everyone in the Wiley Lab who have been super, super useful, particularly Carolyn for the collaboration that we had, really, really good collaborator, Gina, who's been really useful for scientific discussion. Dan, who's in the lab, but super helpful. And everyone who read and talked to me about it. I grew a lot in every aspect of my life, primarily because of interacting with members of the lab, and Marvin, of course, huge pillar of the lab. I would also like to thank my family. Gracias por todo. And to the Georgia Tech community in general. It's been really, really awesome being here, and the Center for Microbial Diseases and Infections, really, really awesome learning and discussing, and yeah, thank you all for being here. And with that, I will take any questions that you guys may have.
All right. Well, thanks, Juan. So we can take questions from the audience, and the committee members can hold off. You can just anyone chime up, or if you're too scared to chime up, you can put it in the chat and I'll read it.
I can ask a question to get us started. Nice job, Juan. That was really great. And
thank you.
I was just thinking when you were talking, especially when you were talking about comparing the Pseudomonas and staph interactions in mixed and in static communities. I was wondering about kind of the gradient within there. Like, there's a lot of difference between like the environment not moving at all, which is probably, I don't know if that's even totally accurate. Like in the host, is there some movement going on versus like a mixed, heavily stirred environment? And you've kind of been looking at these extremes, but have you ever thought about kind of looking at the in-betweens too, and how could that help you figure out like when spatial structure is important or the mechanisms that dictate spatial structure?
Yeah, there was this paper by Gary Borisi recently where they were doing spatial structure, looking at microbes' spatial structure in the gut, which is sort of like an in-between because there's constant flow, and he described it as unmixed chemical reactor. And so, yeah, I think that is quite challenging because you have, I think, incorporating the other dimension, which is time, would be interesting in doing. It would be basically characterizing the spatial structure across spatial dimensions, but also across time, and see if, as you mentioned, some of these properties are like emergent properties. So how this evolves through time and through space would be interested. In regards to how relevant these superstirred systems are, or what is the flow on something that is not a gut, I think there's pretty limited, especially because aggregates tend to not move that much. They're not motile, and because they're so large, they're bulky to move. So I would think that there's some flow, and I see that in the microscope sometimes because they're like drifting. But I think I think incorporating the time dimension would be quite useful.
Yeah, that makes a lot of sense.
I don't know if I answered your question.
Let's go to the next question because we still have 10 minutes. Okay. You can get that through. Yeah. Shada, you had a question?
Yes, thank you. Thanks, Juan. It was really nice talk. It was great to see all of your work in one presentation. I have a question about some of your unfinished work on the tissue that you were imaging from the chronic wounds and some of the abscess model that you're working on, especially for the chronic wound ones. What I was surprised was you have like around 200 micrometer in depth in your Z-stack, if I remember correctly. You don't see a lot of human cells, or not human, tissue in terms of like mouse tissue in blue color, or I don't know if the way that you're presenting it, we don't see it. You don't see a lot of mouse tissue.
Yeah.
Do you think it's because of the model, or do you think it's because of the processing after? And do you think you're going to use like some hydrogels in future to kind of like keep the tissue intact, so the imaging would be easier, or something like packed analysis to get rid of the background and keep the tissue intact?
Makes sense. So the to answer your question, I think it's a combination of basically all of the above. I think some part of the the DAPI, we don't really wash them. Some of the instructions are that we wash them, but if I wash the DAPI, then it would be disturbing the spatial structure. So some of those things are overexposed. The other issue with this is that if you think about it, I image them immediately, right? So I image within 20 minutes, I am already imaging. And so what I think too is that there's not a lot of time for the DAPI to go in. Penetrate. Yes. And honestly, because I know all of these things, the analysis has been, I haven't even looked at DAPI. The DAPI for me is just background, is just sort of orienting myself. But because of these issues of maybe I'm overstaining, maybe I'm understaining, I still don't really know. I just, to me, it's just,
it's just very interesting.
Really, some cases it looks really clear that, okay, this did it really well, but sometimes it's everything's blue completely. And then, And in regards
that's very interesting, because you're trying to set it up, and I know that it's not finished work. So it was interesting to know what are the challenges. And it would be really nice to optimize it, because then you can have different type of tissues. And if you want to use markers for inflammatory responses, then you can do that as well. But that's very great. Thank you.
Yeah.
All right, James, you had something?
Yeah, great talk, Juan. Quick, simple question, hopefully, right? So you said that the Pseudomonas is moving away from the staph under your HHQ condition stuff. By what method is it moving away? Do you know what form of chemotaxis is actually happening there?
I'm sorry, I should have misspoke. I don't believe I cannot show that they're actively moving either away or towards them. What I can tell you is that I showed an enrichment distance, that the spatial organization is different. In regards to the specific mechanism, is completely unclear. I am assuming that there's some sort of chemotaxis going on. I can assume that in regards to staph is not even motile, right? So maybe it might be. Yeah, I did see Diggle's talk on that. But yeah, I don't believe, I cannot tell you for sure that they are actively moving towards or away from anything. What I can tell is that they form this different spatial organization in response to multiple differences in the environment. Now, there's some talk about I can speculate about it being cues that modulate this, but and some inhibitory.
What about just what about just knocking out the pili or the flagella and seeing if you have a flagella mutation or a pili mutation, does it still do it? Is it actual motility, or is it just enrichment and growth in that area?
That has been an experiment that we've been wanting to do, tried motility mutant. But in regards to the abscess, both of the microbes are non-motile, right? And so you can see that and motility is something that is, that isn't in a short span, and it's pretty random. So, and these patterns, for me to see a signal, these have to be pretty much embedded into the system, right? And so, as I mentioned before, they're largely aggregates, and aggregates aren't specifically known to be motile, right? So you can see that this, I don't think that actual active motility is a main driver of this behavior, because I see it in non-motile organisms, and because aggregates, which is largely what I see, are largely non-motile. So this might be other kind of partitioning of the environment.
Oh, sounds great. Thanks, Juan.
Yeah.
All right. So you have one from Kelly. Hey, Kelly. Kelly has a question for you, Juan. It says, do you have any ideas for why PA's HQNO makes SA more tolerant to TOB? And do you think it's triggering a general antibiotic defense, or is it a specific interaction?
Yeah, I think this has been studied. It's specific to TOBRA. Well, it's been shown that it's specific to tobramycin, and yeah, what I believe is sort of like a general response to sort of shutting down anything that's coming from the outside, and that's why it's become more tolerant to tobramycin, essentially.
All right. Do we have any other questions from the audience? I don't see any more in the chat. I don't think I do. Okay. Well, sounds good. Well, thanks, Juan, and thanks everyone for coming. And the committee can stay on, and everyone else can sort of take off, and then and the real grilling begins. That was a good turnout. It's over 50 people, so that's a lot for.
Yeah, it's a great turnout. Yeah.
Yeah, it was a good turnout, especially with a lot going on.
Yep. Yep.
Yeah, I agree. 50 is pretty huge.
Juan drinks beer with a lot of people.
I mean, there's also a lot of my family here.
Awesome. That's cool.
You can drink beer with your family. It's fine.
I certainly do.
Juan, you can actually, I think you're a moderator, so you can get in and just start kicking other people off if they don't, if they're not off.
If you want, we could go to a breakout room and have our discussion while you say goodbye to your family.
I don't...
But as moderator, you'll have to send us.
Go in the apps.
In the apps, meeting highlight, whiteboard, breakout session, start, and then...
You can assign people there.
Okay.
Just assign us.
Yeah.
So, is Brian phone is the same? Is that you, Brian, or no?
Oh, send me both there. I was having weird glitches with my internet, so I'm listening on my phone and watching on my computer.
Cool. Marvin.
That's all those indirect costs paying for that Georgia Tech infrastructure, huh?
Just Jerry Emerson, Peter. Something to look forward to. We call it
the IT guy.
It's all the radium, man.
So I think I sent you guys the invitation. Or did you get it, Sam?
No.
There you go. There you go.
Okay. 5, 4, 3. I'll see you soon.
Okay. ¿Qué onda, ma? No te oigo, estás en mute. No te estás en, no sé cómo. Hay un micrófono. Déjame, veo si te puedo quitar el mute. Ahí está. No puedo, no te oigo. Pero bueno, si quieres, luego platicamos. Dale. Bye.
Yeah.
¿Qué onda, pa?
Híjole. Oye, muchas felicidades, Juanito. Increíble. ¿No?
Gracias. Gracias.
Ya, mención honorífica, señor.
No, pues todavía no termino, todavía no sé.
Esa es la defensa, hay otro evento.
No, bueno, o sea, es la defensa y ahorita son 5 profes que están en el comité, que están en el panel que están en mi comité, que son los que firman la tesis, ¿no? Entonces, a ellos les mandé el escrito, que te digo, fueron como, es básicamente escribir todo. Escribí 5 capítulos de la tesis, ¿no? Y luego, sí, más bien para la audiencia, pero bueno, ahí lo platico. Ya te tienes que salir, señor.
Ok, muchas felicidades.
Gracias, pa. Bye.
Bye.
That's awesome, man. Is that your grandpa?
No, that's my dad.
That's your dad? Oh, awesome. Shit.
Yeah.
Yeah, cool.